Okay, good afternoon, everybody. Thank you very much for coming. My name is Malcolm Brown. I'm president of the Geological Society. And welcome to this, the third Society London lecture of 2017. The lecture is entitled Engineering a Dinosaur, Using Mechanics and Engineering Analysis to Understand Feeding Behaviour and Skull Evolution in Dinosaurs and Other Extinct Animals. And it's my pleasure to introduce our speaker, Professor Emily Rayfield of Bristol University. How fossil animals lived, moved and fed has fascinated scientists and the public for decades. The fossil record provides us with behavioural clues, from remnants of the skeleton and other hard tissue remains, and the occasional exceptionally preserved fossil. Bite marks, stomach contents, coprolites, tooth shape and skull form offer us direct and indirect evidence of extinct animal feeding ecology and behaviour. However, many outmoded behavioural inferences and just so stories remain unchallenged and untested. Using a combination of computational paleobiological methods, such as CT scanning, and then applying biomechanical principles and methods drawn from engineering structural analysis to understanding the feeding function of fossil vertebrates, Professor Rayfield will show how these digital methods are reshaping the way we can conduct paleobiological science, focusing on dinosaurs, fossil mammals, and the earliest tetrapods to occupy land. And that is the longest sentence I've read out for quite some time. Emily Rayfield, a paleontologist at the University of Bristol, researches this very subject. In particular, she uses mechanical principles and finite element analysis to understand the function and behaviour of the animal skeletons, living and extinct. Ladies and gentlemen, please welcome Emily Rayfield.
Okay, thank you very much for that introduction. So today what I'm going to be talking to you about is research that involves looking at engineering methods to understand more about how dinosaurs functioned and more about how they evolved as well too. If I can... There we go. Okay, so I noticed that this year the Geological Society London lectures are on the theme of risk. And there are going to be a few elements of risk in my talk here, mainly to do with the risk of fracturing your bones. And also, again, partly risk of actually facing down an animal like this 66 million years ago would be perhaps quite a challenging situation. And it's this kind of feeding behaviour in animals, and dinosaurs in particular, that have captured the imagination of the general public and scientists as well too, in movie and film and TV programmes and things like that. And that's led to these kind of more scholarly reconstructions of feeding behaviour in these animals. But a large part of what my research involves is actually looking at whether there is any kind of scientific basis to these kinds of scholarly reconstructions. So, as mentioned in the abstract of the talk, there are a number of lines of direct evidence that we can look at to give us some information about what dinosaurs were actually eating back in the Mesozoic. And one of those things is gut contents. Exceptionally preserved fossils like this animal here. This is actually a dinosaur here called Microraptor. It's feathered. It's one of the more derived theropod dinosaurs, very close to the origin of birds. And what's significant about this fossil is it's found with the remains of fish in its gut cavity here, which gives us some direct evidence that these acid-etched remains were part of the meal of this last meal of this animal. We have other evidence as well from exceptional gut contents. This is another theropod dinosaur, and within its gut region here were the remains of not one but two different types of animal, the hind leg of another dinosaur, and also the remains of a very early bird called Confuciusornis, also found preserved within the gut cavity of this animal. So this gives us some good direct evidence what these animals may have been eating. Another line of evidence, as I mentioned in the abstract, are coprolites, or fossilized feces. And this is perhaps one of the biggest coprolites that we know of, as you can see by the scale bar on here. And it's in the exact time and place that gives us a clue that it was made by the animal Tyrannosaurus rex. And this particular specimen has been sectioned, and inside are the fragments of broken and shattered herbivorous dinosaur bone. And that tells us not only that these animals were being eaten by large carnivorous animals, but also that those bones were being fractured during the process of ingestion, and also, because they're not very digested, that the gut passage times were also relatively fast as well too. Other evidence comes from things like bite marks and broken teeth on specimens. And at the top here, we've got a theropod tooth. You can see it here, and it's embedded in the vertebrae of a sauropod dinosaur. Here we've got the pelvis of Triceratops. And in here, where these black arrows are these puncture marks, sometimes associated with these striations as well too, which are the right size and shape to have been made by Tyrannosaurus rex teeth. We also frequently find things like scratch marks here, which are indicative of bite scrapes along the bones here. We see these as well. Here are some ribs with also these characteristic scrapes along them. And this is an example of the one that got away. So here we've got an X-ray of the vertebrae of this hadrosaur dinosaur here. And that bright structure there is a theropod dinosaur tooth, which has become embedded in the vertebrae. And then around the bottom here, we can see that the bone has healed and grown around that tooth. So this animal actually survived this putative attack. So these give us some nice kind of lines of evidence about what dinosaurs were actually eating. Tooth size and shape is also extremely helpful. So classic carnivorous-shaped teeth here, like these recurved teeth here from small tyrannosaurs in this case. They often have sharp edges on the front and back, and these serrations which can vary in size and shape. Sauropod dinosaur teeth are often peg-like, with the wear on them differentially placed in different species of sauropod dinosaur. These animals were herbivores. And also we get a range of different tooth types of varying complexity in some of the ornithischian herbivorous dinosaurs. So these leaf-shaped teeth here are similar, the teeth that we find in things like ankylosaurs, these heavy armored dinosaurs with big club-like tails. The leaf shapes are very similar to the teeth of some modern herbivorous lizards. But we can see very complex teeth, in this case in ceratopsian dinosaurs. This is the group that includes Triceratops. And these are found together in these elongate tooth batteries, which would form quite efficient processing surfaces. And this is taken to the extreme in animals like hadrosaurs, where they have these individual peg-like teeth, which are jam-packed together in these very broad tooth batteries. So also we can get clues not only from the teeth, but from the shape of the bones themselves. So the neck anatomy of dinosaurs can give us a clue as to how high some of these herbivorous animals could reach into the canopy, but also how flexible the necks were horizontally gives us a clue as to the feeding envelope of some of these animals. Also as well, we can build muscle reconstructions which give us a clue as to the flexibility of necks. For example, in this case, whether these carnivorous dinosaurs used their necks in a manner similar to modern-day raptorial birds while they were feeding. We also can infer the ecomorphology of dinosaur skulls as well, so simply using the form or the morphology to infer the animal's ecology. And you can see here from this range of different skull shapes, we can infer that basically they were built to do different things. We have elongate skulls here, which wouldn't be able to close their jaws very quickly, but wouldn't be able to close their jaws very forcefully, but would be quite quick. Also, we see losses of teeth. We see some other forms here with slightly shorter skulls here, where the leverage would be more advantageous. And so a variety of different shapes here, which we can infer have different functions. And this is exemplified in modern-day mammals, for example, where we see this whole range of different skull forms. So things like toothless anteaters, baleen evolving in whales, bats which can eat nectar, these high-crowned grazing teeth in things like horses, the slicing carnassial teeth that we see in carnivores and things like that, and the associated changes that we see in the shape of the skull. So one way that we can actually characterize and quantify differences in shape is using a landmark-based method called geometric morphometrics. And essentially what we do here is we identify a series of homologous landmark points in the same place across different skulls or different parts of the skeleton. And then we can build up an idea of the variability in shape of those different features. And this is what's been done on this chart here for different types of theropod dinosaur skulls. And where we see these individual dots here represent different skulls, and where those dots cluster closer together, that means they are similar in shape. And what the results of this study showed, that these skulls were you were more likely to have a similar shaped skull if you were closely related to each other than if you ate similar things. So what does the shape of the extinct animal bones therefore tell us about their function? So we can kind of think about this in a sort of hierarchical system like this, where we kind of start off with the mechanisms here which dictate the shape of the animal's bones. So it's genetics, developmental processes, and the environment in which it lives. And this in turn then gives us a picture of the shape or the morphology of the skeleton. And from this then, if we're using this kind of ecomorphology principle, we infer from this the feeding ecology of the animal in question. But you can see from this chart here that we're actually missing out a whole series of intermediate steps. So one of those being the kind of mechanism by which the morphology creates. So if we think of the morphology as being the bones and the associated soft tissues, the mechanism is how these bones and soft tissues work together to create a series of kind of hinge, levers, and pulleys, which work as a force transmission system, for example, in the skull or in the limbs or whatever, to enable the animals to feed or to move. And this mechanism, therefore, operates a particular function. In the case of the skull, these hinge, levers, and pulleys operate to open and close the jaws. And we can measure the effectiveness of this function by looking, for example, at performance metrics. So, for example, as the jaw is opening and closing, how fast is it? Or what kind of bite force is the animal exerting? Or how much stress and strain is it building up within its bones when it's undertaking this kind of behaviour? So we can think about morphology and try and infer ecology, but what we're missing out is this middle step in here, which is how the whole morphology actually works, how the system works. So we can look at not only what did dinosaurs eat from direct evidence and skull shape and tooth shape, but we can also look more in a more detailed way about how did dinosaurs eat. And this can answer more broader questions about how does the shape of the skull relate to the function of the skull, and how have those form-function relationships evolved over time, and therefore how are those different systems functioning. So if we look at this then, what kind of information do we actually have preserved as fossils versus what kind of information do we really need to answer these kinds of questions about how function really works? So we obviously have bones. We have fossils preserved to varying degrees of completeness, and we can use these as the basic building blocks to infer how the skeleton might have functioned. And in fact, actually, it's been known for well over 100 years that the shape of a bone reflects the loads that it experiences during function. So this is a diagram of an idea that was first proposed by Julius Wolff in 1892. And he noticed that the orientation of trabeculae in the head of a human femur matched the orientation of the major lines of tensile and compressive stress in a bent cantilever beam, bent in such a way where it's loaded at the top like this, in the exact way that the head of the femur would be loaded. And what he suggested was that these trabeculae within the femur were actually aligning themselves along these major axes of stress within the femoral head. And therefore, the bones had modelled itself in response to the loads that it was experiencing. So there's a relationship, therefore, between functional loading and form. So the way that this would actually work is outlined in this diagram here. So we start off on the left-hand side by having some kind of externally applied force. So this could be the animal biting into something, it could be a ground reaction force as it's running along. And this loading then exerts a stress on the skeleton itself, with stress simply being the load per unit area. And this in turn, depending on the material properties of the structure, would generate strain within that bony tissue, a stretch per unit length. Now in extreme circumstances, this would lead to deformation. But this is sort of a feedback loop here, which the amount of stress and strain is controlled by the material properties of the bone and also the structural organisation of the bone as well too. So the material properties being things like the Young's modulus, so how elastic or stiff the tissue is, its density, something called the Poisson's ratio, so its tendency to change in width when it's stretched. They all impact on how stress and strain are received within the skeleton. And also the bone then can reorganize itself at various different levels to withstand the stress and strain. So like Wolf said, it could orientate trabecularly along these lines of major stress, or it can build more tissue in a region which experiences large amounts of stress. So we get this kind of system where the bones become what's called mechanically adapted for the functions that they experience. So we have this kind of signature of function in the skeleton. Now, this has been modified, as you might expect, since 1892, since Wolf put it forward. And this is kind of put together in this system in the 60s by someone called Harold Frost, who described something called the mechanostat. And this has served as a purpose for people to actually look for evidence of this in bones themselves ever since. And basically, he suggested that most bony tissue operates within what's called the adapted state. So if you or I were walking down the street, we wouldn't be experiencing loading on our bones, which will be in any way out of the norm. But for example, if our bones, for some reason, fell into disuse, the everyday strains that we would experience in the tissues would drop, and therefore we would lose bony tissue because it simply wasn't needed to withstand those loads. Whereas if the bones were overloaded, for example, taking up a new running regime or something like that, then tissue would be deposited and bone would be gained. And there's actually many lines of evidence to suggest that this really is the case. So, for example, if you take up running in middle age, there's a study that shows that your bones become more mineralized. People like ballet dancers have much thicker and stronger bones in their legs than non-ballet dancers. And a good way of looking at this is kind of in paired studies. And a great example of this are sportsmen and women who use one arm or leg preferentially to the other. Tennis players are a great example. The muscles are bigger in their racket-holding arm, and therefore they exert more load onto the bony tissues, and those bones are therefore bigger as well too. And a very interesting study showed that greyhounds that race around the track, because they tend to race in the same direction every time, have thicker bones on one side of their ankle than on the other to represent this running around the track in the same direction. So we've got these very practical examples of how bone tissue can adapt to changes in the loading regime. And there's also very obvious examples of bone disuse, so bed rest and weightlessness, which is a big problem during space exploration, where tissue falls into disuse. And also another study showed with our greyhounds here, these are my retired greyhounds here, that once they actually stop racing around the track, the bone tissue remodels itself, so they lose, they say, symmetry in the tissue deposition within their ankle bones. So it shows that remodeling is still taking place. There's other studies that have been done kind of in a more experimental framework, for example, loading or unloading tissues in different types of animals and looking at the response. And it's also now established that mechanical loading is really important for normal skeletal development as well too. So these are two mice embryos. One's normal on the left and the other one on the right-hand side has got a mutation, meaning it doesn't have any muscle tissue. So it's not impacting any loads onto the skeleton. And as it proceeds through development, we see an increasing abnormalities in the skeleton. You can see it's got a curved tail. The ribs haven't formed properly. There's problems with the skull and the head as well too. So mechanical loading is really important at all different stages of life. Also, some of our studies have shown that if you eat different things, you have different shaped jaws. So this is a study that I was involved with where we had lab mice and they were raised on different diets. For 38 weeks, one group had normal mouse pellets, which we classified as hard food, and another group had these mouse pellets which were ground up and mixed with agar jelly here to be the same nutritional value but softer food. And what happens is these hard food and soft food eaters after 38 weeks have got statistically different shaped jaws. And also, the hard food eaters have got jaws that are much more robust. They're deeper where the tooth row is. And these processes here where muscles attach or where the jaw joint attaches are more well developed. And we thought this is interesting because it suggests that actually their jaws are adapting better to deal with harder foodstuffs. And we did a series of kind of biomechanical analyses that showed that this actually was the case. So the hard food eaters have got what we call more efficient or more functionally advantageous jaws. They have better leverage and for different muscles and also biting at different teeth. Also as well, the hard food eaters' jaws are more resistant to bending and twisting. And then the hard food eaters have got lower stresses in their jaws when they're feeding as well too. So not only can eating different things give you different shaped jaws, it can also give you jaws that function in a more advantageous way for what you're actually eating. And they're adaptations that all go within the individual lifetime of an organism. But actually we can think about this on broader evolutionary timescales, which is the kind of thing that I'm interested in when looking at fossils. So for example, a change in shape can then lead to a change in function, which can then lead to kind of different adaptations for different purposes within evolutionary history of organisms. So to summarise that then, what we've got is that the behaviour of organisms and their bits and pieces are governed by the principles of physics and engineering. And so we can apply physical principles to determine how the structure might have functioned and interacted with its environment. And we're lucky because in deep time the animals and the environments might have changed, but the laws of physics haven't. But also, really importantly, the bits and pieces, the cellular and the tissue level building blocks that build skeletons and muscles remain unchanged. And as far as we can tell, so do their material properties. So what we're interested in then, and what I'm particularly interested in, is measuring functional stresses and strains within bones, so that can give us a clue to the functional purpose of those tissues. Now, one way we can do this if we have living animals is to use methods called strain gauges. These are little resistive elements that you place on the surface of a structure, and they measure changes in strain. This is fine if you have a living animal, but it's not brilliant if you have a fossil and a skeleton that's turned to rock. We can use mathematical approaches to work out stress and strain, which work very well when we've got simple structures or simple material properties. When the structure becomes more complex in shape, that maths becomes much more challenging. So that's especially so if we think about the skulls of animals, which are extremely complex geometries. So one way we can approach this is using an engineering technique called finite element analysis. And this is a technique where we can deduce stress and strain within a structure. And it basically works by taking a complex geometry and breaking it down into a finite number of individual blocks called elements, which have a simple geometry for which we can calculate the stress and strain within those elements. So for example, in our structure here, we've got 3 cube elements here, which are joined at their corners by nodes. And what we would do is we would give that structure specific material properties. We've fixed it at one end, and we've loaded it at another end with a particular magnitude of force. And the analysis will then calculate how much these nodes are displaced in response to this applied force, taking into account the shape of the structure and its material properties. So it basically gives us a way to calculate stress and strain within complex geometries, like animal skeletons. Now, this method has been used for many, many years in engineering analysis. This is a car park and a plane and a geological feature, nuts and bolts, shoes for some reason. And also it's been used a lot in the last kind of 10, 15 years in zoology and paleontology for that purpose I've just described to you, which is looking at stress and strain within skeletons. And how to read these kind of very pretty colour maps is that where you see hotspots here is where we have large amounts of either stress or strain or displacement, whatever metric is being looked at here. So the way the process works is that you run through this kind of series of steps, which are the exact same steps that you would do if you were making a model of a car or a bridge or a nuts and bolts, which is basically starting off with creating a digital model of the structure that you're interested in this case a dinosaur skull, breaking it down into a finite number of these individual elements, giving it material properties, and then applying some loading conditions that mimic the function that you want to look at, and then analysing the results, so looking at stresses and strains and deformation. So I'm going to take you through how this process works in a dinosaur skull. And this is the skull here, as it's still in the ground, of a late Jurassic fossil, Allosaurus fragilis. You might be able to just make out the shape of the skull there. There's the dentary, the lower jaw. Here's the upper jaw up here. And this specimen was when it was found, was largely complete. This is an assembled cast here, which is in the Wyoming Geological Museum. And what we did with this specimen was put the skull through a CT scanner. Now, CT scanners, you may be familiar with, are ways in which we can get X-rays which look not only at the external surface of structures, but also give us a view of the internal features. And they work in the same way for human bones as they do for dinosaur bones. You just need more energy to get through the rock. And this is a picture here of a slice through the CT scan of this dinosaur skull here. And you might be able to make out here it's got these lacrimal horns up here, and we're cutting through in this individual slice through these horns here. The light areas are bone and the darker grey areas are matrix. So we can build up a whole series of slices through the animal's skull like this. We can identify different features. We can even look at the shape of the brain, for example. But we can use this as the basic information that we need to build a 3D model of our dinosaur skull. And we use this to then break it down and create this finite element model here of the dinosaur skull. So this is basically a three-dimensional mesh of this structure. One of the things that we need to know, though, if we want to run this analysis, is how stiff the bone is. Now, of course, these tissues have been lithified. They've been turned to rock, so we don't know how stiff it is. But luckily, actually, bone material properties don't really vary that much across a whole spectrum of living animals. And so from that inference, we can say that probably in extinct animals, they were pretty similar. And what we see here is a thin section through some bone in the skull of an Allosaurus here. And it's got some features like lots of remodeling and turnover of tissue that we see in other large herbivorous mammals, for things like cows, for example. So it seems to have a sort of a signature of a large animal kind of bone material properties. So this is what we used in this model. We then need to think about the soft tissues. Now, ordinarily, soft tissues are not preserved. So how do we manage to work out what the muscles were doing and where they were attaching in the dinosaur skull? Now, one of the things here is that actually where muscles attach to bones, they usually leave a mark. And this can be in the case of like a little ridge or a depression or a scar of some sort. And so we can use this as evidence for where muscles might have attached and inserted into the bones of the skull. And we can also look at living animals. And if we look at crocodiles, we find actually the muscle arrangement in dinosaurs isn't massively different to the muscle arrangement that we see in crocodile skulls as well too. So we can use this as a guide. And that allows us to build up these 3D pictures of where the muscles may have sat within the animal's skull. And once we know how big a muscle is, we can work out and estimate how much force that muscle can generate if it contracts. Muscle physiology, again, a bit like material properties, is fairly conserved across vertebrates. So we can use that as a guide. So we can simulate biting, for example, by loading the teeth at different points along the tooth row. We can simulate a force at the jaw joint as the upper and lower jaws are brought together. And we can think about how those different muscle forces, where they attached, how much force they were generating when they were contracting, to build a picture of how the whole structure works. So the results of analysis look something like this. So in the bottom left down here, we have a plot here where the yellow areas show where we've got increased compressive stress within the bones of the skull when the animal's biting. And these stresses form trajectories around some of these openings in the skull here, which these struts and arch seem to act like the pillars of an arch, constraining this load within there. And this diagram here on the right-hand side shows where we see peak stresses within the skull, particularly in the skull roof up here at the top. Our calculations of muscle size and muscle force allow us to make an estimate of how hard the animal could bite. And if we put this in context, what we find is that our dinosaur here, Allosaurus, is biting somewhere in the realm of a lion or a leopard. So perhaps not particularly hard. And animals like T-Rex, for example, are way up here at the top with some very extreme examples of biting. But what we can also do is, because we're looking at this from an engineering standpoint, we can look at the safety factors within the structure. So basically the ratio of the everyday load to the highest load the structure can experience before it breaks. And what we find within the dinosaur skull is that actually it's operating at a safety factor of between 10 to 26, depending on different ways you can parameterize the muscles. So there's a lot more bone there than is needed simply to withstand the biting force. So The lower jaw, though, is much weaker, has a much lower safety factor. So what this means then, perhaps the skull is subject to some kind of compromise constraint, meaning it's much more strongly constructed than it needs to be because it's serving a different purpose, holding the eyes or the brain or something like that. Maybe our estimations are just wrong, which is possible, but maybe also as well what's happening is that there's more dynamic forces that are taking place here, that the skull is actually more over-engineered to withstand increased impact from biting forces generated by things like the neck, for example, rather than just the animal clamping its jaws together. Okay, so these forces may have come from, for example, the neck helping skull-prey contact and things like that. So we can also use these models to look at the effect of comparative stresses within different types of dinosaurs. So here's our Allosaurus model here. Here's some models of our simulated stresses within the bones of a smaller theropod dinosaur, Coelophysis. And then also here as well, the Tyrannosaurus rex as well, a much larger animal. And you can see that these stresses are following similar trajectories to the skull. The skull is stressed in similar ways in compression and in tension as well too. It's being pulled apart on the bottom and it's being compressed upwards towards the skull roof. But there are a few subtle differences between different groups, and this allows us to pause for thought and think about how they might relate to differences in the bones that we see. So particularly we can see this in the back of the skull here in the allosaur. This is where the highest amount of compression is occurring up here at the top. And this is kind of in line with the skeleton itself. But at the back here are two bones, the frontal and the parietal, which are really tightly fused together. Further forward here are some elongate bones called the nasals, which are paired on each side and kind of loosely connected together. In contrast, what we see in Tyrannosaurus is that those peak stresses are occurring further forwards in the animal's skull, here and here, for example. So they're moving away from the back of the skull, and they're occurring in the region of the nasal bones. And it's been a long-standing kind of puzzle to paleontologists, people who study dinosaurs, is why tyrannosaurs had these really big, thick, fused nasal bones. And what this analysis suggests is that it's because of the stresses that it's experiencing during the skull are occurring exactly where we find these nasal bones here. So there's a link there between the skull stress and the way that the bones of the skull are constructed. So that was kind of one of the earlier studies that I looked at in terms of building how the method actually works. But I'm going to talk to you a little bit about two other studies now looking at beaks and looking at Jurassic mammals as well too. And the first one is focusing on the evolution of the avian beak. And the bird beak is a kind of classic example of adaptation. It formed the foundation of Darwin's theory of natural selection. And we see these range of beautiful beak shapes in different types of living birds as well too. And the evolution of the beak, a number of ideas have been put forward as to why the beak, or what may be the advantage of having a beak over teeth, for example. One of these is to do with weight saving or mass saving. As these animals were evolving flight, a beak was lighter than having teeth. The other one is to do with food procurement, that a beak was actually more effective at grasping and processing seeds, which appear to have become an increasing part of early Cretaceous and other Mesozoic bird diets. So one of the things we can do is actually look at beak evolution. And beaks are interesting because they're not only found in birds, they're also found in a range of different vertebrates, things like turtles, for example. But they're also found in other theropod dinosaurs on the bird lineage that clearly were not flying. So one of these groups that are fascinating are the therizinosaurs. These are quite a small group in terms of species, quite poorly known animals, which ranged in size up to very extreme kind of 5 to 7 meters or so in height. This particular animal here, Earlykousouris, is from Mongolia. It was a smaller therizinosaur. By smaller, I still mean about 2 meters or so in height. But it was one of a few lineages of clearly non-bird theropod dinosaurs that evolved a beak. So by studying animals like this, we can get an idea about what was the role of the beak in these non-flying animals, and what difference does having or not having a beak make to skull mechanics. And we can also test the idea about whether the beak reduces the mass of the skull as well too, whether it serves some role in these animals. So this is the skull of Earlykousouris here. It's the only complete skull of a therizinosaur dinosaur that's in existence. There are a few lower jaws, but this is the only skull. It's, as dinosaur skulls go, it's really nicely preserved. And we CT scanned this in Tring in Hertfordshire, where a company that makes CT scanners are actually based. And here's the skull sat in the scanner here. And here's the Mongolian scientist Palaeontological who found this specimen, actually holding it while looking at the results of the CT scanning on the screen here. And what we're able to do from this scan is to build up these really nice resolution 3D digital pictures of the skull itself. And we can orientate these in different ways and things like that. And you can see from this specimen here, it's interesting because it's got teeth at the back of its jaw here and also along here, but at the front, the teeth are absent. And these little holes here along the surface of the bone are the kind of structures that we see in animals that have a beak. So they suggest that there was a beak across the top here. We see these same kinds of features in, say, turtles, for example, as well. So that suggests to us that there's no teeth here. There's no evidence of teeth anywhere, and that there was a beak sitting over the top here. So this was work that was done by my then PhD student, Stefan Lautenschlager, who was very talented at doing some of these digital reconstructions. And what he's been able to do here is actually fill in some of the gaps by mirroring bones from different sides digitally to create this complete 3D reconstruction of the skull. And Stefan's very adept at making these kind of reconstructions of what the animal may or may not have looked like. And you can see here, this black area here is one of our estimations of where the beak might have sat, based on the absence of teeth and also these little sort of foramina that sit along the surface of the bone. So again, using the same principles that I talked to you about before, identifying where we see little ridges and scars on the bone, we can work out where the muscles may have attached. And then from that, digitally, we can build up these chunky 3D muscles, which allow us to measure the volume of those muscles, in which we can then use that to estimate how much force they could generate when they contracted. And here's some of our cross-sections through the reconstructions of where these muscles may have sat in the skull. So what we found when we looked at how much bite force these muscles could generate, and these are our estimates down here along the bottom, they range in value based on our top and bottom estimates of muscle physiology, but essentially it's somewhere between about 40 to about 134 newtons. And so to put that in context, this is about the mean range that a 3 to 6 year old child could generate. So if you've ever been bitten by a 3 to 6 year old child, you know it's quite a forceful bite, but it's nowhere near the kind of large bite forces that we would see in things like Tyrannosaurus rex and that kind of thing. So from that then, what we were able to do is to sort of create a few hypothetical digital scenarios of what might be going on with the extent of the beak. So we've got our 4 different models here, with the star representing what we think is our best guess estimate of how big this beak was. So here in our model, we've got a big bit of keratin that's sitting over the end of the jaws here, and here on the lower jaws. And we've got one model up here at the top, which is missing any beak at all, a big beak model, and then a model which has had teeth put in as well too. And from this, what we did was estimate how much extra mass having a beak would actually give you. And these are the results here. And what we find is that actually having a beak is not really saving you that much in terms of mass compared to actually if you have teeth, at least in this animal here, which has particularly tiny teeth, which lots of the very early birds would have as well too. We're also able to look at what effect the beak has on the mechanics of the skull. So these very dazzling colour plots here, essentially what they're showing you here are the stresses within the skull and the displacement within the skull, how much the bones are moving. And this is for our 4 different simulated models, lacking a beak, little beak, big beak, and teeth instead. And this is for bites at the front, the middle, and the back of the jaws, and repeated. And so the key things here are to look at the difference between these colour plots between animals which don't have a beak and animals which do have a beak. And by eyeballing this, what we can see is that it looks as if the stresses are higher in animals that don't have a beak, and the displacements are higher in animals that don't have a beak compared to the ones that do have a beak. And we can do various methods that I'm not going to go into in great detail, but to actually quantify and get numbers out of this analysis, which shows you here that this is kind of the maximum amount of displacement, so how much the skull's moving, from our original model with no beak compared to our model with teeth in purple, compared to the green and the red, which are the models with beaks. And so those green and red values, in most cases, are lower than the other two values, which shows that having a beak lowers the amount of displacement that we're seeing in the bones. So our conclusion for this was that before appearing in early birds, we see beaks in these herbivorous dinosaurs, which are clearly not flying, and the presence of the beak seems to be helpful in reducing the amount of stress and strain during feeding. So the final example then I'm going to go through is looking at Jurassic mammals, which are from fissure-filled deposits in South Wales and the area just around Bristol as well too. So these are the fissure deposits around Ogmore and St Bride's, from classic kind of quarry deposits like Ueni and Pontallen and places like that. And these are carboniferous cave type environments which contain these deep fissures and caves into which early Jurassic and late Triassic mammals and lizards and dinosaurs have fallen into, and their remains have been preserved. And two animals of particular interest to us for this study were Morganeucodon and Cuneiform. These are very small, kind of shrew-size animals which sit right at the cusp of true mammal evolution. And they have a range of features which link them to true mammals, like having a mammalian jaw joint, and also a number of features which link them back to their predecessors, such as having a trough in the sides of their bones where they have what we call post-dentary bones, which we, as true mammals, have either lost or have incorporated into our middle ear as accessory hearing ossicles. So these are really important animals in terms of mammalian evolution. This is work that was done with a collaboration with my postdoc, Pam Gill here, who's for many years has studied these early mammals from the fissures. And here's some reconstructions of Morganeucodon and Cuneiform here. The jaws are about the same size. They're quite tiny, that's 2 millimeters there. And they're quite similar in overall shape, but they have a few key differences, especially in Morganeucodon. The back of the skull here is much more developed, where muscles attach, and it's got a more developed jaw joint. Its teeth are broader as well, with these broad kind of cusps, compared to Cuneiform, which has got a less developed rear lower jaw and more pointy teeth here. And for many years, it's been suggested that these differences in morphology and in tooth shape suggest that the animals ate different things. But simply beyond that, we can't say anything more than that. So we were interested in this challenge because Pam knows the animals really well, as to whether we could actually extract any more information out of these specimens. And the interesting thing is that actually when you find them in the fissures, they look like this. So they're disarticulated, jumbled blocks of bones. The white bits are bones in here, by the way. But the nice thing about this is that actually the individual bones themselves are preserved really well. And you can basically take these lumps of fissure material, dunk them in a bucket of water, perhaps with a bit of calgon as well too, and it helps break down the individual bones so we can extract them really easily. What we then did is use very high-resolution CT scanning techniques, either a micro-CT scanner or synchrotron radiation scanning, which runs from a particle accelerator, to get really, really high-resolution 3D images of these fossils. And from that, what we could do was stitch together a digital reconstruction of the jaws, which you can see here looks very similar to those outline line drawings. But what we've got here is all the internal information as well too. So we were able to use this then in our engineering analysis to think about whether there was any functional differences between these two jaws. So in the same way that we'd done before, we thought about where the muscles might have attached, how big these muscles might have been, and when we simulated these muscle contractions on our two jaw models. We also looked here at the leverage of the jaws themselves, and we calculated something called the mechanical advantage. And this is basically how efficiently the jaws can open and close. A higher value for mechanical advantage indicates a more forceful bite. A lower value indicates a less forceful but a faster bite. And so these two differences here between Morganucodon and Cuneiform, although the jaws are similar lengths, these numerical differences suggest that this is more forceful and this one is faster. So then our finite element simulations also showed big differences in the way that stress and strain was accommodated within the skulls as well, within the jaws. So we simulated the amount of muscle force it would take to generate a bite of 2 newtons. And we use this particular value because this has been shown in studies of living animals to be the amount of force it takes to pierce through an insect cuticle. So something that realistically a shrew-sized early mammal would be eating. And what we find is that the maximum stresses within the jaws are very different, over twice as high within Cuneiform than they are in Morganucodon, even though these things are about the same length. And the maximum stress at fracture of modern-day bone is between about 150 and 200 megapascals. So if we think about safety factors, Cuneiform biting at 2 newtons is operating pretty close to what would be the extreme value of force that it could actually generate. Whereas Morganucodon very comfortably could bite through something with 2 newtons of bite force and still keep the stresses within its jaws relatively low. So that gives us some good clues that they were functioning in different ways. But what we were really interested in was whether they were actually eating different things. And this is where we combined our functional analysis with some studies of the tooth texture and some quantitative, what we call tooth microwear analysis. So this is an SEM picture at the top of the Morganucodon tooth and a Cuneiform tooth. So these are teeth from the same fissure, the same point along the tooth row, and the same facet. And these 3D textural maps show very different results. The teeth of Morganucodon are much more ridged. You can see where these red lines here, than the teeth of Cuneiform, which are much smoother. So this suggested that those teeth were the textural mapping was very different, which suggests that they were eating different things that were roughing up their teeth in different ways. So one way we wanted to know really what they might have been eating was to actually do a similar study on modern day animals, which we know what they were eating. So we got some tooth textural analysis from different species of British bats. So these are horseshoe bats up here at the top. These animals tend to have a much higher proportion of hard foodstuff in their diet, things like beetles. This is the brown long-eared bat here. This has a higher proportion of much softer foodstuffs in its diet. And then down here we've got the common pipistrelle and the soprano pipistrelle, which is a kind of cryptic morph of the common pipistrelle, which just differs because it has a different type of song, different type of vocalization. And these eat kind of a range of mixed diets. So what we did was we did the same kind of tooth textural analysis on the teeth of these modern day bats, and we found that they had very different roughness parameters in their teeth. And the teeth of Morganucodon were most similar in terms of their tooth microwear patterns to the teeth of the greater horseshoe bat, the one that was eating lots of beetles and hard foodstuffs. And our Cuneiform here had a tooth texture that was more similar to the brown long-eared bat, the bat that was eating much softer food items. And we can statistically represent this in this plot here, where we have our modern-day bats in these different coloured regions here, where the data points are statistically distinct from each other. And this chart basically shows here, the further you plot along this axis, the more kind of intractable or hard prey you have in your diet. And Morganucodon largely, those triangles here, plots in this hard foodstuff area. And Cuneiform, these are from different fissures, plot in this soft foodstuff area down here. So not only were these jaws functioning in different ways, our studies also suggest that they were eating very different things. And this is interesting not only from in the case of these two individual animals, but it shows that really early on in the history of true mammals, these animals were not just generalized insectivores like we always thought they were, but they were becoming functionally and ecologically quite diverse. And also they were doing this before true mammalian characteristics had been fully established in mammalian evolutionary history. So it gives us quite an interesting perspective on kind of the way in which these animals are actually evolving. So that was the last example that I was going to give you there. There was a wrap-up slide there. So basically, just to kind of go over a little bit about what I said, was that, we've got fossil bones, and it's been established that they do adapt their shape to deal with different functional loads. And they can do this over lifetimes, and they can also do this on evolutionary timescales as well, too. So we can use that information to our advantage. So we can use things like digital imaging and 3D reconstruction of techniques, and use biomechanical and engineering analysis to sort of test hypotheses of function and adaptation in fossils. Things like the feeding performance of large carnivorous dinosaurs, the functional and mass-saving significance of beaks in bird lineage dinosaurs, and then look at evidence for previously unrecognized functional and ecological diversity in past ecosystems as well too. So I will leave it there, and then thanks as the customary thanks to all these people who've helped fund this research, and I'll hand it over to you for any questions.
Fantastic. Goodness me, what can you get out of bones? And teeth, particularly. Questions? Two chairs, yeah. There's a microphone coming. If you just wait for the microphone, please. Thanks.
Not knowing enough about paleontological biochemistry, I don't know if the hormones driving these male and female animals were similar to mammalian. So whether there was an estrogen effect on the bones of females, and whether there's any evidence of osteoporosis or osteomalacia in the animals.
Yeah, that's a really interesting question, and I can tell you from more actually has been known about that actually in dinosaurs because of turnover within the bony tissue during egg laying. And we see similar kinds of turnover during periods of egg laying in birds. They turn over the calcium and the phosphate within the bony tissue when they're laying eggs. And there's been evidence for this kind of deposition of medullary bone, kind of intermediate bone tissue, which sort of acts as a brief store of calcium and phosphate, then that's actually removed away during periods of egg laying. And we see that in some dinosaur bones. So there is kind of a hormonal signature there within those dinosaur bones. Within the mammals themselves, I can't think of anything specifically that links that directly in some of the fossil mammals, shall we say. But certainly within dinosaurs, we see similar changes to bone structure that we see in modern day birds as well too during these egg laying cycles. So kind of a hormonal control there.
The fisher mammal reptile mammals, there seems to be some differentiation in the bone pattern, more from, particularly in one, there's a change in the teeth pattern from the front going rearwards than in the other. That ties in, I would think, with what you've been suggesting on the basis of wear and strength of bite and so on. Is that so?
Yeah, so Morganeuton, the animal that was at the top in that reconstruction there, has got more what we call heterodont dentition. So it's got sort of molars, and then it's got a small canine, and then it's quite procumbent incisors, so sticking out at the front. Whereas there's less of that differentiation in Cuneiform. And certainly the shape of the molar teeth is more, and I think the presence of the incisors and the canine, especially for kind of puncturing and capturing prey, would tie in a bit more with that. But Cuneiform is a bit of an enigma because looking at it, you would think it actually is less derived than Morganeuton, but actually it sits in different places within the tree when we try and place it in there. And I think we have good jaw material, but we just don't have any postcranial material as well, so it's difficult to place it. Yeah.
Thank you.
Just wait for the microphone, thank you.
Is it possible to do the mechanical analysis to look at how the head is involved? With it, you said the safety factor was too high, or does it just get too many? Yeah.
So what we can do is actually do that in combination with something called multibody dynamics analysis, which actually looks at the movement of the muscles as well too. So if we can actually bring in that kind of analysis that looks at muscle action with the stress-strain analysis, we can do a combination of those things together. And actually, the picture that I showed at the start of the allosaur dinosaur with the muscle reconstruction, that was a group in the US who've actually done that. And they actually showed that it, from what I was saying from the stress analysis, did seem to be borne out by the muscle reconstruction as well, that they were using the neck quite extensively in feeding as well too. Yeah.
Last question at the back.
Thank you for a fascinating talk. Not knowing much about this subject, but I think I've read say modern pandas have much bigger teeth than is necessary to eat bamboo shoots, which seems to be their complete diet. Could this have happened in prehistory as well? You know, you can't simply assume teeth shows what they were eating.
Yeah, yeah, exactly. Yeah. And I mean, and that's the polar bear, I think, as well, is another example of that. That its teeth, when you put it into these kind of broad scale analysis, suggests something completely different as well too, depending on what you look at, how you look at the teeth. So I think with the mammal stuff, what we were interested in doing was trying a few different lines of evidence to see if they all suggested the same thing, to give us a bit more confidence in our results. And so we did the engineering analysis and the microwear analysis, and then also there was some other work that we did as well too, doing some more kind of standard biomechanical tests too, which also supported the idea that Morganucodon was different to Cuneiform, and it was more robust, and it had a more forceful bite, and it was eating different things as well too. So I think the way forward is to bring in lots of different lines of evidence to help with that kind of problem. Yeah.
So last question? Go on, one more.
Maybe a second bite.
Sure.
Quite at the very beginning, you were referring to contents of gut contents and coprolite contents indicating diet. I referred to bones being cracked, passing through rapidly, presumably no sign of acid digestion, suggesting a short gut or a different gut chemistry.
Yeah, so I think that's a really good question actually. I think there was some signs of acid digestion, but not much, I think. But I think the throughput times must have been pretty quick, because the bone could be actually identified almost down to the family level by the actual structure of the material that was in there. So it was certainly, it was quite easy. It was a hadrosaur dinosaur. And given what was around in the environment at the time, it was probably Edmontosaurus or something, because that was, it was most likely that was actually the case.
Very good. I think we're just about out of time. So if you could join me again to thank Emily for a fantastic talk. A question I didn't ask. Yeah.
Thank you very much. I've always got more questions. Can the DNA teeth or is it
There's a lot of debate around what you can actually see in terms of textures. There's been a few recent studies that suggested that you could get some proteins out of dinosaur bone. There's other lines of work that suggest that you can see blood vessels, even with blood cells inside it as well. But that's quite disputed. They do look like blood cells.